So it's 4 p.m. Good afternoon and welcome to today's bioMérieux webinar with the title Low Endotoxin Recovery Assessment of PDA Technical Report number 82 and Demasking of Endotoxin with Dedicated Sample Treatment. My name is Christian Faderl. I'm the project coordinator of Endotoxin and LER services. I'm based at Heigloss in Germany, which is the endotoxin unit of the bioMérieux. I apologize for my bad voice today, and I hope that the voice will stay with me until the end of the seminar. We would like the session to be interactive, and we invite you all to participate by sending questions and comments at any time via the question function on the right, which you find on the navigation bar. After the presentations, we will have some more time to answer all of your questions or most of your questions. This
Christian, you're going to have to start again. Sorry.
Okay. So it's 4 p.m. Good afternoon and welcome to today's bioMérieux webinar with the title Low Endotoxin Recovery Assessment of PDA Technical Report. Endotoxin and LER services. I'm based at Heigloss in Germany, which is the endotoxin unit of bioMérieux. I'm sorry for the bad voice today. I hope that the voice will stay with me until the end of the seminar. We would like the session to be interactive, and we invite you all to participate by sending questions and comments at any time via the question function on the right in the navigation bar. After the presentations, we will have some more time, some more minutes to answer your questions. This webinar will be registered and is also available for reviewing afterwards. So we will have two talks today. The first part of the webinar is about TR82, low endotoxin recoveries takeaways. In the second part, I will present you the ENDO-RS technology and bioMérieux LER services. So I want to introduce the first speaker, Kevin Williams. He's an expert in the field of endotoxin detection with more than 30 years of hands-on experiences in the area of pharmaceutical quality control. Kevin Williams is well known as author as well. So his new book with the title Endotoxin Detection and Control in Pharma, Limulus and Mammalian Systems was published recently. So I pass the ball to you, Kevin. Welcome and thank you that you participate this webinar.
Thank you, Christian. And thank you for joining this morning. I hope you can see my screen. I have to apologize in advance. My voice is a little gravelly from getting over a cold, so please bear with me. I'd like to talk about
I can't see your screen. Sorry, Kevin.
Oh, sorry about that.
Now it's working. Thank you.
Okay, thank you again. I'd like to talk about Technical Report 82 on low endotoxin recovery. Slide is not advancing. Okay, oh, there it goes. Sorry about this, little technical difficulty. The last year, the PDA published Technical Report 82 on the topic of low endotoxin recovery. It's an important reference for these kind of studies, 120 pages, 23 authors, experts in the field that cover the expectations in performing hold time studies. It provides an extensive background on BET in general and lists some methods of mitigating masking in biologics. Here specifically, I want to overview the mechanism of LER, talk about the importance of biologics process control and hold time studies, look at 9 important categories governing the hold time studies from the TR82, a little bit about NOE masking results, and 12 case studies. These will not be gone into in depth, but you'll be able to see what's there for further reference. I'm having some trouble advancing the slides. There we go. Okay, what is LER? We remember that in 2013 in Genentech, Chen and Venter published a paper that described the specific drug formulations that bring about the masking of endotoxins in solution. We see that when a chelator and a surfactant are put together, these in turn act to dissociate the biologically active monomer or aggregates, the micelles, so it loosens them up, and where the surfactant can get in and coat the individual monomers. So each of these phenomenon, the chelator, that can be overcome, of course, surfactant, that can also be overcome, but the effect of LER is much more profound as a time-dependent formation that is not easily reversed. So it's good here at the start to differentiate control versus detection. Control are the thousands of things that you do to maintain microbiological contamination control, aseptic processing, filtration, all these unit process steps that you go through. And detection runs parallel to this. Detection is done in parallel in terms of both bioburden and endotoxin. On the next slide, we'll show why. And the problem with masking is that at some point in the process, when these formulation constituents are combined, you end up with a solution that cannot be tested with Lumulus-based tests. Of course, the finished product release test is a long-standing QC expectation. People often ask, why can't we just test bioburden? And I think the best reference for this is the BPOG study on microbial monitoring for biological drug substance manufacturing. This makes it clear that between the various downstream processing steps, these are non-sterile filtration steps. They're also pH neutral, so they're fair game for microbes to grow in. Whereas the bioburden, the actual living organisms, can be filtered out step to step. Endotoxin, of course, cannot be filtered out due to the size of the product, the protein molecules. And here's a quote from this report. A too numerous to count result for any in-process bioburden sample should be automatically result in an action level or out of spec result. And you can see this is not overly stringent because any number just under TNTC could be present in the process and not justify such a out of spec. Now, protein masking has not really been addressed for 20 some odd years. It just didn't ever get the attention that LER obtained, but it's now being addressed as a factor in LER per the technical report. We see this chart from Pesh. He tested a variety of proteins on the left-hand side, and these are just proteins in a buffer. You can see the charge on the protein as evidenced by the isoelectric point from 11 to 8.5. So these are cationic. So these positively charged proteins will bind endotoxin, which has a negative charge, and you can see the initial EUs per mil there tested in LAL. Most of that was masked when it was tested after addition of the protein. So the binding of the protein negated in some cases 98% of the endotoxin detection. He was able to reverse that by using proteinase K So on the far right-hand side, you see the original numbers are pretty much unmasked. This is a list that I found in the reference at the bottom. This shows that most monoclonal antibodies are cationic proteins. Some 3/4 of these will be expected to have that positive charge, and you could expect some masking from these even without the LER effect. Okay, this slide speaks to the hold time study as required for a BLA submission. The purpose of the hold time study, quoting from the TR82, is to determine whether a product has LER under conditions which are relevant for the manufacturing process. So in the so-called reverse study, the original drug product is spiked at day 7, and then you spike at 4, 2, 1, 4 hour, and 0. So you're working yourself backwards to the test date where it all can be performed on the same plate. This is a way to reduce the variability, and this is the way that's recommended, not required, but recommended at this time. Of course, the hold time study is critical because it's required for the biologic license application submission. If masking is found, mitigation will be required to be resolved during submission or after approval as part of a post-marketing commitment. FDA is requiring 3 lots to be hold time study tested with a write-up and submission using process relevant conditions, which I'll talk a little bit more about. And as a heading for the remainder of this presentation, we'll look at 9 categories covered in TR82 and 12 case studies. So beginning with the 9 categories to consider for hold time study performance, reagents and materials. They want you to use reagent sets from qualified vendors as part of the BET 85 expectations. The spiking of an undiluted sample, if lyophilized, reconstitute the vials. Spike using CSE or RSE. The quote there is that these are used as worst case analyte. The spike should not exceed 10% of the total volume. You don't want to water down the LER effect artificially. The spike level should account for sample dilution according to the validated method. So you're going to use your QC method. Of course, you can change that and revalidate your method, but the good place to start is to use the method you have. And you'll want to allow for recovery somewhere around the midpoint of the standard curve. The storage container should be the original container or other container appropriate for BET. So the TR82 did relax that initial requirement a little bit that you're allowed to use other containers. Batch requirements, 3 batches used to capture sample variability. That's pretty common for most kind of studies. Storage time and temperatures. The study should be conducted at process relevant temperatures and times. These are the times and temperatures of the process likely to impact the phenomenon. This would be a part in the process where the 2 process constituents that lead to LER are combined. So basically here it's saying that you want to know how long the final drug solution is expected to sit in the LER solution conditions before QC testing. Of course, the goal is to have a QC test that can detect endotoxin. Common hold time temperatures are referred to as ambient or around room temperature and refrigerated temperatures. Something I thought was interesting was reference to a high concentrated endotoxin preparation. It says that you can use it, but it should not be used as a theory for calculation. So if you use it, you'll want to use the value obtained as the starting value. But also you can use the theory for the CSE or RSE as an appropriate value for reference recovery calculation. Continuing with the 9 categories to consider, the controls, a spiked LAL reagent water control should be included, spiked and diluted to the same products, the same as the product samples. Equipment requirements are equivalent to BET. The personnel, there should be some training and documentation to perform these kind of studies, just to show that they've done it before and they can adequately perform it. Data analysis, compare the water control with the sample recovery over time. At bioMérieux, we also use the positive masking control to show that the expected masking actually happened. There are some rare cases in which the masking doesn't happen and you know something was wrong with that part of the study. The TR recommends 4 time points included over 7 days, and the pass-fail determinations based on 2 consecutive less than 50% recoveries. No averaging of time points, and the sample time 0 recovery should not be used to adjust the recoveries. Okay, the time and temperature of the whole time study should be process relevant. The quote is, refers to process steps most likely to have an impact on LER. For example, the addition of polysorbate plus chelator hold times, open or close process steps. And you can see here, they say if there's a process relevant step conducted at refrigerated temperature and 1 or more performed at room temperatures, the study should be conducted at room temperatures. So this is a worst case philosophy again. Okay, so we performed the hold time study. We've got some data from the 7-day hold of the direct spike. So how do we know if we have masking? It should become fairly obvious if it's dropped down. And also we can get some idea of the strength of the masking effect by looking at the masking kinetics. So you can see the green line is barely going down over the course of time, whereas the red and yellow lines are more vigorous masking effect. This will give some idea of the difficulty of mitigating this phenomenon. Naturally occurring endotoxins, I know some believe that this is an artifact of the purified endotoxin, but there's now many studies that have shown the naturally occurring endotoxin is susceptible to LER. And here's some endotoxins from grown up naturally occurring organisms, E. coli, Enterobacter coaki, marcescens. The only organisms in this small batch that were not as subject to LER were the Pseudomonas type organisms, or what used to be Pseudomonas cepacia and Pseudomonas piquetii. The rest of them were clearly subject to LER. Okay, we'll look at a very short overview of the 12 case studies that are included here. And you'll have to remember that this is probably a couple or 3 years old by now, even though it just came out last year. So some of these studies are probably not as progressive as you would expect if you perform 12 studies today. But the hope is here that you may see something relevant depending on what your product issue is, and then you can consult the TR82 directly. So case study number 1, polysorbate content relative to protein content. So they were looking at a monoclonal antibody, drug substance, 120 milligram per mil, and a drug product, 90 milligram per mil. So these are pretty high protein concentrations. The protein in this case, and it would be specific to the protein, of course, in the absence of the polysorbate did not show LER. But there was a correlation between the protein content, which affected the LER with the same amount of polysorbate. So the protein certainly makes a difference, even if it's not causing the phenomenon. Case study number 2, evaluate the storage conditions post-spike, 50 milligram per mil protein, citrate, polysorbate 20. This risk assessment that they perform provides a means of defining risk categories and levels and suggests mitigation approaches. So it looks like that this is a complex proposition involving a process change to change the process in order to get a test faster before the LER phenomenon can take over the sample. Number 3 and number 4 were both synthetic LER solutions with some NOEs. Study 3 showed that the majority of crude NOE preparations also suffered LER-based recovery loss. Number 4, temperature, pH, salt concentration using the same synthetic LER solutions. They got some help with the phenomenon by diluting in magnesium chloride. We'll see that again. Number 5, they explored different ways of calculating the percent recovery. They showed that the starting amount of endotoxin, of course, is critical because that's what you'll be comparing against. They studied different ways of calculating it. I don't recommend messing with that. I would recommend just keeping it simple. Number 6, the masking and resolution in a MAB. Here's a successful treatment using cation replacement buffer, half a molar magnesium sulfate and 1 molar tris. This method was able to provide satisfactory recovery studies of CSE. So this is something to bear in mind. Ruth Daniels, I think, has a paper on this that you can Google. Case study 7, this is the one that Christian will be describing in some detail after this presentation. Develop sample preparation protocol for a problem sample matrix. The ENDO-RS method has been used with many molecules now to demask endotoxin from different species. And this protocol used both reference standard endotoxin and naturally occurring endotoxin to show that regardless what the contaminant is, you're going to find the endotoxin. Number 8, investigate and overcome LER driven by protein alone. They designed a reverse hold time study and introduced specific hold times based on the data. Here again, it looks like they're going back to the process and adjusting the process, which might not be applicable to most people at this stage of the game when you're looking for a QC test. Case study 9, LER case study for another monoclonal, high protein monoclonal with chelator and surfactant, and also histidine. Here they were able to simply change the LEL, and this change of LEL formulation gave a better recovery. So this is something simple to try first. If you can switch your LEL to one that allows your hold time study to pass, then that's a method that would be easy to validate the change in LELs. Case study number 10, the CSE recovery for drug substance with LER constituents. Here again is a risk-based in-process control, which involves a process change. So I don't know how open manufacturing will be to such process changes, but it's certainly something that can be looked at and correlated with the hold time study. Case study number 11, no LER effect in sodium phosphate polysorbate formulation matrix. So here is one that you might on paper think that there would be some LER, but this is a fairly low protein concentration, 5 milligram per mil, and this compound did not show LER. Number 12 is in the same boat. It doesn't say what the protein concentration there is, but also they were able to recover. So just because you have the theoretical possibility of LER, it's going to depend on the hold time study to tell you whether your recovery is acceptable or not. So finally, a summary of the 12 technical report case studies 3 mitigations via sample treatments, 1 with ENDO-RS that Christian will discuss, and 2 using a cation buffer magnesium sulfate or magnesium chloride. 1 simple LEL change, 2 risk assessments. There may have been 3 risk assessments of the process. 1 process change, 2 molecules that did not show LER, even though they had the potential or the theoretical potential for it, and 3 studies based on synthetic solutions rather than the actual product. So these were exploratory samples. So I hope this gives some background as to what's out there in the TR. You may not have had a chance to purchase or read through it. So if there's something here that piques your interest, then please either follow up with us or go directly to the TR82. At this point, I thank you for bearing with me. My voice is giving out a little bit, and I'd like to turn it over to Christian. Thank you very much.
Yeah, thank you, Kevin. I will take over. Okay, thank you very much. So, in the next half an hour, I give you a deeper insight into the bioMérieux LER service and the ENDO-RS demasking kit. As Kevin mentioned already, I will also present the case study 7 of the technical report a little bit in more detail. Those who have already dealt with the topic of LER and demasking know that this is a very complex phenomenon and many things influence the masking and demasking of endotoxins. In order to get as much experience as possible about demasking, bioMérieux cooperates with 2 excellent laboratories, Microcode Biotechnologie GmbH in Europe and ENDO Nutrisciences in the US. All projects performed in these centers of excellences are under supervision of bioMérieux, which supports the whole demasking project process with the experience of bioMérieux endotoxin R&D team. Besides feasibility studies for the RFC-based endonext assays, the endotoxin service portfolio of bioMérieux is mainly focused on LER. We offer whole-time studies, in some initial demasking screenings, and the most important part is, of course, the endotoxin demasking service. Every sample, every drug product is different, and therefore all behave different regarding masking. Of course, we gained a lot of experience in the demasking during the past few years, and we see some similarities when working with similar products, but nevertheless, the demasking project is always unique and a unique research project. Here you can see the project structure, how nearly all our demasking projects look like. This is just an example, and of course, it can vary from project to project and from customer to customer. First of all, there's the whole-time study where you determine that you have an LER issue, and then you enter to the milestone 1 in the endotoxin service project. In this milestone 1, it's the root cause analysis. What is the cause of the LER? Is it the active pharmaceutical ingredient? Is it the formulation buffer or the process relevant containers? And here we test also some quick demasking approaches, as Kevin presented already, like dilution in magnesium chloride or something like that, which is mentioned in the technical report as well. Then we enter in the milestone 2. This is the biggest part, the development of the demasking protocol. Usually this second milestone ends with the evaluation of the robustness of the development of this demasking protocol. There we check different parameters. For example, we check the inter-assay precision, linearity, lot-to-lot experiments, and so on. To make sure that the customer can implement the demasking method at the end, we train the customer on the demasking protocol in the 3rd milestone of the project. In this method, R&D method transfer or training, We invite the customer to come to our lab with some employees, that they get a hands-on training how to do the demasking with the ENDO-RS kit, and how to implement this then in their lab. And also we go to the site of the customer to train them there and make sure that the demasking is working. Here are some estimated timelines for each milestone. Again, this is a demasking project and it's a research project, and so the timelines are just estimations. Could be that the development is very fast, could be that it takes 9 months before having a stable demasking protocol in hand, could be that it's less, of course. Apart from the behavior of the drug product, the project timeline depends also on the test conditions. For example, endotoxin limit plays a major role, the masking time, and most of all the detection system, so the detection assay you use. If we develop a demasking method with endolyzer, or when we develop a protocol with the LUL assay, the times we need for getting a stable demasking protocol can vary a lot. Most often, the development with the endolyzer detection assay is way faster than development with a LUL. The endolyzer is an RFC-based assay with a special sample preparation, which I will show you in more detail later on. All demasking projects are under supervision of the bioMérieux R&D and toxin team. One project manager will work on no more than 2 demasking projects, and approximately 2 full-time employees will work on the project per month. Our demasking tools cover the common tools which are described in the technical report as well. For example, addition of divalent cations, dispersants, and some other sample treatments. Usually these methods were used by the customer already or cannot solve the demasking issue. So for complex samples, we always need and use the ENDO-RS kit for demasking. So the ENDO-RS kit is a sample preparation method for endotoxin recovery in biopharmaceutical formulations. Originally, it was developed for samples in which the masking is triggered by surfactants such as polysorbate. But it can be used also for demasking of samples in which proteins have a huge influence in masking. The kit is universally usable, so to say. Because it is just a sample preparation, an endotoxin assay is still needed. Possible high content of the added demasking reagents can lead to interference of the LUL assay and RFC reagents. Therefore, we highly recommend, especially for unexperienced users, to use the RFC-based assay endolyzer for detection. So what is the procedure for the endotoxin recovery with the ENDO-RS kit? First of all, the samples have to be spiked with a certain amount of endotoxin. This should be done according to the guidelines in the technical report 82. After the incubation or masking time, the samples are treated with the ENDO-RS components, and the demasked sample is ready for endotoxin detection. This way, the spiked endotoxin will be detected completely. After sample treatment with the needed amount of ENDO-RS reagents, the LER effect should be overcome at any time point of the whole time study. The goal is to get an endotoxin recovery around 100%, of course. So on the left side, you can see how a whole time study can look like with an LER effect. And on the right side, there is shown in green how a LER study or a whole time study should look like after treatment with the ENDO-RS kit, after treatment with the sample preparation, which was developed to demask the samples. The demasking kit is a toolbox. There are 6 components with different functions in there, which can be used for demasking. So there's a buffer for the pH adjustment, there's a disturber to destabilize the LPS masker complex. There's an adsorber in there to bind surfactants, 2 modulators which should support the reconfigurator, and the most important part of the kit, so the key component of the ENDO-RS kit is the reconfigurator E, which should stabilize and lead to aggregation of the LPS to make them visible and detectable again. Which components are needed for the endotoxin demasking depends again on the sample. The components are added to the masked endotoxin in the drug product in alphabetical order, and then this leads to demasking of the LPS and the formation of the detectable structure for factor C based assays. The demasking procedure has 4 main steps. There's the initial screening, optimization of the selected demasking approach, lowering the endotoxin level to the desired endotoxin limit, and then possible further optimizations. In the initial screening, a higher amount of LPS should be used for spiking in order to determine the most useful and needed ENDO-RS reagent combinations. So, for example, 100 EU per milliliter or 50 EU per milliliter should be used for this first screening. After the rough component screening, the concentrations of the components should be optimized until the demasking results is satisfactory. In the next step, the endotoxin amount should be lowered stepwise, and the developed demasking schemes should be evaluated for these endotoxin levels. At the lower amounts of endotoxin, further optimization experiments could be necessary, or optimizations have to be implemented so that other optimizations have to be used. For example, dilutions, incubation times, and so on. So this is how an initial screening scheme could look like. One example is given also in the package insert, but it is just an example how this can look like. Of course, it's important what your product is, what your formulation is, and then you have to adjust the components according to that. Due to differences in the buffer compositions of these mask samples, the amount and concentration of each component of the required demasking reagents must be adapted, of course. Here's a more detailed example for a first screening table. So there are different combinations of the ENDO-RS components which lead to different recoveries. For example, in the approach 1, there will be added component A, component B, and component E in different dilutions of the component E, and then on the right you can see the percentage of the recovery of the demasking. The best recoveries were obtained here with the last demasking scheme and the highest dilution of component E. This scheme should then be used for further development at the lower endotoxin amounts, or if this is already at your endotoxin limit, then just make some repeating experiments to see the robustness of this method. So as I showed before, with the developed demasking protocol, the endotoxin recoveries should be always between 50 and 200%. The ENDO-RS was developed for use with the endolyzer as detection method, but it can also be applied prior to LUL, which requires a sample-specific interface optimization. The big advantage of the endolyzer is the special sample preparation step, which makes it way easier to see demasked endotoxin. The endolyzer uses the common RFC reagents, but there's a special pre-coated 96 well plate included in the kit. Sorry. Because of the ELISA-like format of the analyzer kit, it is commonly used for very difficult samples which interfere common RFC or LAL assays. Endotoxin binds to the fake proteins located in the wells. The interfering substances can be washed away and the bound endotoxin can be detected via fluorescence. And this is, of course, very helpful for demasking. Additional to the drug product and the excipients in there, there are also the added ENDO-RS components in the solution. And depending on the concentrations and the interactions, this mixture can interfere the test. And this can lead to additional masking, for example, or can destroy the demasked endotoxin. Using the analyzer kit, the demasked endotoxins, they bind to the plate, and all ingredients are washed away, and the endotoxin will be detected without any more issue. So this is the biggest advantage here in the analyzer kit. So the combination of the ENDO-RS and analyzer will lead to faster progress in your demasking project. The combination with LAL is possible, but linked to a more time-consuming effort. So even if the RFC assay is no option for you in the quality control test with the analyzer, then you can see if the demasking is really functional or not. So to say, to use the analyzer, it's the door opener for your demasking project. So, and such a procedure to use the analyzer for a door opener is described in the technical report 82 in the case study 7. All these information are from the technical report itself. So in this case study, a demasking protocol development using the ENDO-RS kit is described. The sample contains citrate and polysorbate, so it seems to be a classical LER effect here. Endolyzer was used initially in this project to gain a starting point, as I said, and then the protocol was transferred to a LAL detection assay. Finally, this ENDO-RS components were used to do the demasking. So they used the components A, B, D, and E in this case study. For the evaluation of the robustness, several experiments were performed. 21 independent runs with an overall variance in recovery of just 20%. So tests of different incubation times, vortex times, and lot-to-lot experiments were included here, or the sensitivity, so how low can you go with the endotoxin level, and even the demasking of different LPS were shown here. All recoveries were in a valid range of about 100%. So it's a very nice example for the use of ENDO-RS for endotoxin demasking. At the end, I want to give you some takeaway messages. Use the tools at your disposal to overcome the LER. You can also use the ENDO-RS kit. And when you don't find a demasking solution, contact us. bioMérieux has a demasking service. And for any questions regarding endotoxin service and/or support, please contact me, and we will find a demasking solution for you. So thank you for your attention. And now we have some time for questions. The questions will be forwarded to me. This will take some moments. Thank you. So here we have some questions. The first question, how do you deal if you submitted a product and you discovered afterward that masking is possible on the product? Yeah, Kevin, do you want to address this? So first of all, you should the FDA, if you submit a new BLA to the FDA requires a whole time study. So you need to do a whole time study first, of course. So for all new products which were for all new products, you will need to do a whole time study. Kevin, do you want to comment?
I'm sorry, I got disconnected for a brief moment and I didn't hear the question.
Okay. How do you deal if you submitted a product and you discover afterwards that masking is possible on the product?
I think so. Initially, you communicated to FDA that there was no masking, and later you discovered masking. I think that you're obligated to inform them of the update because at some point they're gonna insist on seeing the data.
Thank you. So another question, any recommendations for using NOE in whole time studies in place of RSE, and any articles defining the use of RSE? So in the technical report, it's described that you should use for whole time studies the CSE or the RSE. NOEs were not accepted for these applications for the FDA. You can use NOEs in whole time studies, of course, for just for information. Do you want to add something, Kevin, on this one?
Yeah, I think it's important to develop your test with CSE or RSE and then to check it with NOE, because in the end, what you're looking for are natural contaminants. So CSE or RSE being a worst case should allow for the detection of those without further work, but it's good to verify that.
Mm. Another question, when do you suggest to start considering the LER in the product life cycle? So as soon as possible, of course. So as soon as the formulation is fixed, and so roundabout clinical studies in the stage 2, I would say. So as soon as the formulation is really fixed, then you should first do a whole time study just to see if you have an issue or not, because could be that to develop a demasking protocol could take a while, and when this is then until this is implemented on your side, so it's important to do this as soon as possible. But it makes no sense to start a whole time study when the final formulation is not fixed. So when you still change the formulation, then you have to repeat the whole time study, of course. Another question here, is it allowed to perform the LER testing on an in-process sample before the addition of polysorbate 20? First of all, it's allowed to test LER at any time if you want to. But you should do an LER testing, of course, after adding polysorbate 20, because this is one of the main trigger for LER. Do you have a list of ingredients that might cause LER? You can see whenever there's a chelator or and a surfactant in the formulation, then this will trigger an LER. So this is commonly known. There is a lot of literature now out there which states this, and also in the technical report it's written. There's also an example for masking mechanism, and this is based on a chelator and a surfactant. So when you have always a chelator and a surfactant, it's highly possible that you have an LER effect.
Yeah, and you also want to check particularly high protein concentrated solutions, whether they have any LER constituents or not.
Yeah, thank you. The next question. To calculate the endotoxin recovery of the whole time study, do you compare the positive water control versus the product control? or is it just the spike recovery of the test? So the endotoxin recovery is calculated, so you can calculate to the water control at time point 0, of course, and the theoretical value. This is also recommended in the technical report. The water control at time point X, so which had the same hold time as your sample, is not, it's in some cases you're allowed to use this from the technical report point of view, but you should not use this because there some more effects can occur, like interactions with the vial and so on. Kevin, you want to comment as well?
Yeah, I would just suggest keeping it as simple as possible and comparing your results over 7 days to the initial water control.
So one more question. If we are using the ENDO-RS solution for in-house development studies, will bioMérieux provide guidance? Yeah, of course. We will help you, and we will give some advice what to do, what you can do to overcome the LER issue and to use the ENDO-RS kit. If there are any questions how to use the ENDO-RS kit, we can give advice. So one question, what is the current take on introducing LER pre-treatment in process control bulk samples? Kevin, can you address?
The latest is to start with the finished product, demonstration of your ability to recover spike after 7 days now is the current requirement. And I think the requirements of looking at your whole process are not going to be looked at if you can mitigate the final product testing.
Thank you. So one more question. Have you had a case where it was impossible to find a demasking solution? If so, what did you do? Yes, we had one case, to be honest, but this was not due to a normal LER effect. This was so no biological, and there was, so it was no classic LER, and there we couldn't use the ENDO-RS kit was not good for this one. Yeah, but this was the only one, and it was not the normal protein or citrate or chelator surfactant LER masking. Do you have experience with the registration in EU or FDA with your specific test? So we are not doing registrations with our assay with our solution, of course. So this has to be done by the customer, but we give support, of course. So when a customer has to do has to apply or has to add the sample preparation and register this in the FDA, we give support. And if there are some more experiments to do or so on, or if the report has to be written a certain way, we are doing this, and we are there supportive.
Yeah, most studies thus far have been involved in the post-marketing commitment testing. So to generate this data and show the FDA that this phenomenon can be overcome, because at this point, most people have started rather late in the cycle of fixing the problem. So maybe more and more it'll be addressed earlier. But as it is, most people come kind of late to the demasking testing.
One more question. Can the use of bovine serum albumin and ultrasound help if there is a problem with the critical micelle limit? Kevin, do you want to address this one?
BSA, I believe, is not allowed to be used in drug products. Of course, it's not in the drug product. It would be in the QC test, so that's okay. And there's also human versions of that because a while back, they had the mad cow disease scare, so they don't allow the use of bovine products. But I really don't know if that's going to help the situation or not. To add a different protein than the protein that's already in there may make for a bigger problem. So it's hard to predict, and so oftentimes you say it's empirical. That means you test it and see if it helps.
Thank you. One more question. Have you done any LER studies on clinical placebo? So clinical placebo is normal formulation, or Kevin, can you help me with this one?
It sounds like it's something they give in a blinded study. So a placebo, if it could have an LER effect, there's no protein in there, so it should be easier to mitigate, and also such a process should be, I would think, less prone to contaminants, more straightforward of a process, and probably it could even be terminally sterilized. So I don't know the good answer, but I don't suspect it would be a big problem.
Yeah, so placebo itself, I can say so, not clinical ones, but placebo itself, we did LER studies for this one as well, but so just formulation without API, yes, we did. Okay, then I would close the session. It's nearly 5 o'clock. If there are any questions left which we couldn't answer, please forward this so you can still enter your questions and we can address them and send it back to you via email. Otherwise, please contact me or any other bioMérieux sales rep, and they will forward you to me, and then we can address your questions, of course. So, Kevin, thank you again.
Thank you. Thanks everybody for coming today.
Yeah, and good afternoon, good evening, good night.